Hello, I am Professor Sims, and in this video I will discuss the digestive system. This is the 7th in a series of 10 lessons held as part of my Anatomy and Physiology 2 online laboratory course. If you are a student currently enrolled in this course, please consult the syllabus and the course Moodle site for assignments, quizzes, due dates, and other course information. By the end of this lesson, you'll be able to describe the overall function of the digestive system, the function of the structures of a digestive system, and associated parts on a model, in pictures and diagrams, or in a live specimen. You'll be able to identify and describe the histological structures of the digestive system on a microscope slide, and list the digestive enzymes involved in digestion of proteins, fats, and carbohydrates, and also be able to identify their site of origin. The digestive system is essentially a continuous tube that runs from the mouth all the way down to the anus. Its main function is to mechanically and chemically break down the food that we eat so that nutrients can be absorbed and used by our cells. Within this system, we have the major organs, which include the mouth, where digestion begins with chewing and saliva, then the esophagus, which transport food to the stomach, where food is further broken down by acid and enzymes, the small intestine, the primary site for nutrient absorption, and the large intestine, which absorbs water and forms waste. In addition to these, we have important accessory organs that support digestion but are not part of the main digestive tract. These include the liver, which produces bile to help digest fats, the pancreas, which secretes digestive enzymes and hormones, and the gallbladder, which stores and concentrates bile before releasing it into the small intestine. And together, all of these organs work in harmony to ensure that the food that we eat is efficiently broken down and its nutrients absorbed, keeping our bodies fueled and healthy. Now let's look at the digestive processes in a bit more detail. First, we have ingestion and propulsion. Ingestion simply means taking in food through the mouth. From there, food moves through the digestive tract by two main actions: swallowing, which is voluntary, and peristalsis, which is a series of involuntary muscle contractions that push food along the digestive tract. Next, mechanical and chemical digestion work together to break food down into smaller pieces. Mechanical digestion includes actions like chewing in the mouth, churning in the stomach, and segmentation in the intestines. These all increase the surface area of the food, making it easier for enzymes to work to break them down. The chemical digestion is the enzymes and acids breaking down the complex molecules like proteins, fats, and carbohydrates into simpler forms that the body can absorb. And then finally, absorption and elimination take place. Most nutrients are absorbed in the small intestine, where they pass through the intestinal lining into the bloodstream. Any material that can't be digested or absorbed, such as fiber and waste, moves into the large intestine, where water is reclaimed and the remaining waste is compacted into feces to be eliminated from the body. Let's look now at the mouth. The mouth is where digestion begins, as we mentioned. This is where the mechanical digestion starts via chewing food with the teeth. This breaks food into smaller pieces to increase the surface area, and at the same time it initiates chemical digestion through saliva. Saliva contains enzymes like amylase that start breaking down starches right away. The mouth structure helps in this process too. The hard palate acts like a flap that closes off the nasal passages during swallowing, preventing food from going up into the nose. Salivary glands play a key role in producing the saliva, of course, which not only moistens food, that makes it easier to swallow, but it contains enzymes that begin chemical digestion. And then the tongue, with its taste buds, help us to assess the quality and flavor of food, and also stimulates saliva production and prepares the digestive system for what's coming next. So the mouth is both a mechanical and chemical gateway that prepares food for its journey through the rest of the digestive tract. There are three major pairs of salivary glands: the parotid, submandibular, and sublingual glands. All of these secrete saliva into the oral cavity, helping to moisten food and start digestion, and lubricate the mouth for easier chewing and swallowing. The parotid glands are located just in front of and below each ear. These are the largest salivary glands and primarily produce a watery, enzyme-rich saliva. The submandibular glands lie beneath the lower jaw, or the mandible. These glands produce both watery and mucus-rich saliva, which helps lubricate the food. And then finally, the sublingual glands are found under the tongue. They produce mostly mucus, which serves to keep the mouth moist and help with smooth movement of food. The esophagus plays a role in moving food from the throat to the stomach using peristalsis. As we mentioned, peristalsis is the rhythmic, wave-like muscular contractions that push the food bolus down the esophagus. And at the top of the esophagus is the upper esophageal sphincter, which controls the entry of food from the pharynx into the esophagus. And at the bottom, you have the lower esophageal sphincter, which acts like a valve that prevents acidic stomach contents from flowing back up. And this protects the esophagus from damage. And then together, these two sphincters and the muscular action ensure that food moves efficiently and safely in this one direction, from the mouth to the stomach for further digestion. Here you can see the detailed anatomy of the stomach wall, which is specialized to handle both mechanical and chemical digestion, starting from the innermost layer, the mucosa, which is this entire layer here. This includes the surface epithelium, which produces mucus to protect the stomach lining from acid. And then just beneath the surface epithelium is the lamina propria, which is a connective tissue layer that supports the epithelium, and muscularis mucosa, which is a thin muscle layer that helps to move the mucosa. Beneath the mucosa is the submucosa, and this is the layer that contains blood vessels and nerves that support the stomach tissue. Then you have the muscularis externa, which is this layer, and it is thick and unique in the stomach because it has 3 layers of muscle rather than 2. You have the oblique layer, circular layer, and longitudinal layer. And this layer here, the oblique layer, is like the extra layer that you normally don't see in other organs. This layer helps to churn and mix food in your stomach. Then the outermost layer is the serosa, which is, again, a protective connective tissue covering. Within the mucosa are specialized cells that are important for digestion. The periantal cells secrete hydrochloric acid, which is used to break down food and kill bacteria. And then chief cells, these guys here, produce pepsinogen. Pepsinogen is an inactive enzyme that converts into pepsin to digest proteins. Then you have the enteroendocrine cells. These release hormones that regulate digestion and stomach motility. You can also see gastric pits, these guys here. These are openings on the surface that lead down into the gastric glands where the secretory cells reside. Next we have the liver. The liver plays primarily a role in digestion via its production of bile. Bile emulsifies fats in the small intestine, and it breaks large fat droplets into smaller ones, making it easier for the digestive enzymes to work and allow for better fat absorption. At the microscopic level, the liver is organized into functional units called lobules. You can see those here. Hepatocytes. Hepatocytes are the liver's main cells arranged in plates radiating out from a central vein. So that's what this looks like here, hepatocytes. Between these plates are sinusoids. Sinusoids are specialized capillaries that allow blood to flow through the liver. And as blood passes through the sinusoids, hepatocytes process nutrients, detoxify harmful substances, and regulate metabolism. Meanwhile, hepatocytes produce bile, which is secreted into tiny channels called bile caniculi. These caniculi merge to form larger bile ducts, which carry bile either to the gallbladder for storage or directly to the small intestine to aid in digestion. So understanding the dual function of the liver in both blood processing and bile production, this is important for appreciating the liver's role in maintaining digestion and overall body homeostasis. The gallbladder gallbladder is a small pear-shaped organ located just underneath the right lobe of the liver, and its primary function is to store and concentrate bile produced by the liver. Bile is important for digesting fats, but the liver continuously produces it even when digestion isn't happening. So the gallbladder acts as a reservoir holding this bile and concentrating it by absorbing water and electrolytes. So when you eat a fatty meal, the gallbladder contracts and releases bile into the common bile duct, which then delivers it to the small intestine. And this timed release helps emulsify fats and improves digestion and absorption. The small intestine is the primary site for nutrient digestion and absorption. The inner surface is highly specialized to maximize absorption through several key features. So starting from the inside, the lumen is the hollow space where food passes through. And it's not labeled on these slides, but it is labeled here. It's the hollow space. Then you have the mucosa. The mucosa is folded into circular folds that increase the surface area and slow the movement of chyme to allow for more time for absorption. And then projecting from these folds are the villi. Villi are finger-like extensions covered by absorptive cells. On the surface of these cells are tiny projections called microvilli, collectively known as the brush border. This further amplifies the surface area, enabling efficient nutrient uptake. And then within each villus, there are networks of capillaries and a lymphatic vessel called the lacteal, this guy here. Capillaries absorb nutrients like amino acids and sugars directly into the bloodstream, while the lacteal absorbs dietary fats. And the intestinal crypts between villi contain cells like goblet cells, which secrete mucus to protect and lubricate the lining. Additionally, the duodenal glands, guys here, in the submucosa secrete alkaline mucus to neutralize stomach acid, and the muscular layers, including the muscularis mucosa and the outer muscle layers, help mix and propel intestinal contents during digestion. The pancreas is an important organ with both exocrine and endocrine functions. On the exocrine side, acinar cells produce digestive enzymes that break down carbohydrates, proteins, and fats, and these enzymes are secreted into tiny ducts that merge into the pancreatic duct, which delivers the enzymes into the duodenum, the first segment of the small intestine, where they aid in digestion. Then on the endocrine side, clusters of specialized cells called the pancreatic islets, or the islets of Langerhans. We talked about these in Lab 1. These release hormones such as insulin and glucagon directly into the bloodstream, and they regulate blood sugar levels and are important in energy metabolism. So together, the exocrine and endocrine functions of the pancreas ensure efficient digestion and also glucose balance in the body. The intestinal glands, also called crypts of Lieberkühn, the intestinal glands open onto the surface lining and are packed with cells specialized for absorption and secretion. The absorptive cells have microvilli, just like we saw in the small intestine, to increase surface area and efficiently absorb water, while the goblet cells secrete mucus to lubricate and protect the lining as feces passes through. And then beneath the mucosa, you have the muscularis mucosae and smooth muscle fibers. These help to propel the contents along the colon through the rhythmic contractions. And you can also see lymphatic nodules, which are part of the immune defense guarding the intestinal wall against pathogens. The Lab 7 pre-lab activities are to read the Digestive System chapter, which is chapter 23 in the textbook, review the introduction and background sections from the lab manual, chapter 7, complete 7.1, 7.3 pre-lab exercises, and use those answers to help you complete the Lab 7 quiz on Moodle. The lab exercise 7.1, you're again going to use anatomic models to identify the esophagus, stomach, liver, gallbladder, mouth, spleen, rectum, anus, small intestine, and large intestine. So you have some examples here of anatomical models of the digestive system. So here you have major structures of the mouth and the upper bit of the esophagus. And then moving down below the esophagus, you have the stomach here, liver, gallbladder, the pancreas. There's other things here like the bile duct, the common hepatic duct, the spleen is over here, right? This here is a more detailed model of the stomach with some of the external and internal structures shown. And then this here's the duodenum, which is the start of the small intestine. This here has the small and the large intestine and the rectum here. Then for lab exercise 7.2, you will view histology slides as we've done before. You want to identify the gastric pits, gastric glands, muscular mucosa. There's going to be more than one example of that. Submucosa, villi, intestinal crypts, and duodenal glands. These are some example histology slides. For the esophagus, what you want to do, you'll want to notice first the stratified squamous epithelium, which is this layer here. It's multi-layered tissue designed to protect against mechanical abrasion as food is swallowed, right? Then beneath that layer is going to be the lamina propria, this layer here, and the muscularis mucosa, which is this layer underneath that one. And this is marking the end of the mucosal layer. And then below that is the submucosa, which often contains mucous glands and blood vessels. So we've got a submucosal gland here. This whole thing here is the layer. The muscularis externa has two distinct layers. You have the inner circular and outer longitudinal layers, and those drive peristalsis. And this layer structure helps to move food towards the stomach. And then if you look at the stomach histology slide here, the lining will change abruptly to simple columnar epithelium. So this is the stratified squamous, and this is the columnar, simple columnar epithelium. So this is a key distinction that you will be able to see very clearly at the esophageal gastric junction. In the mucosa of the stomach, you can see the gastric pits. Gastric pits are surface indentations that lead down into the gastric glands. So these pits, all of these pits here. And the gastric glands house different secretory cells like parietal cells, which secrete acid, and chief cells, which secrete pepsinogen. And then the muscularis mucosae lies at the base of the mucosa, this layer here. And then you have the submucosal layer, which is beneath that one. You may be able to see the layers of this. The submucosal layer has the oblique layer, which is unique to stomachs, and it's important for the churning action of the stomach, which we discussed earlier. And then the small intestine here, focus on the duodenum, and then look for villi, the finger-like projections of the mucosa that are covered by simple columnar epithelium with microvilli forming the brush border. So this whole thing here, up here, is the brush border. And as we discussed, this structure is increasing surface area dramatically in order to aid in nutrient absorption. And then between the villi, you can see intestinal crypts, or the crypts of Lieberkühn, which contain stem cells and secretory cells. A key feature to recognize in the duodenum is going to be the submucosal glands. It's not labeled here, but it's these glands here in the submucosa. These are called Brunner's glands, and they produce the alkaline mucus to neutralize stomach acid that we discussed just a moment ago. And you have deeper layers that include the muscularis mucosae layer here, the submucosa, which is this whole layer. This is the mucosa, and you have gastric pits, gastric glands. Then the esophageal gastric junction, that's what we're looking at here. So the esophageal gastric junction, here you want to focus on the epithelial transition. On one side, the esophagus is lined by stratified squamous epithelium. So that's labeled here on this side, stratified squamous epithelium. And then on the stomach side, the other side is going to change to simple columnar epithelium, which is how the gastric pits are formed. So this is the simple columnar epithelium. So this is at the esophageal gastric junction. You have this stratified squamous epithelium, you have the columnar epithelium. There's this big shift right here. You'll see the similar thing in the other side of the digestive system. At the anorectal junction, the epithelium transitions again, where the rectum retains the simple columnar epithelium with many goblet cells, and those are important again for mucus secretion. And then as you move toward the anus, the lining abruptly changes to stratified squamous epithelium, which provides protection as the feces passes through. And in some slides, you may be able to see the squamous epithelium, even though it's keratinized and it resembles skin. But the key thing here is that the transition helps to distinguish the lower GI from the proximal segments under the microscope. So some tips for identification here. Gastric pits and glands, they're only found in the stomach. The pits are shallow depressions. The glands extend deeper. Villi and crypts are present in the small intestine. Villi increase the surface area. Crypts contain stem and secretory cells. The duodenal glands, the Brunner's glands, are only found in the submucosa of the duodenum. Epithelial transitions, which we just described here. So you would look for abrupt shifts in the tissue type, and these are histologically striking and diagnostically useful. Stratified squamous in the esophagus and anal canal, and simple columnar in the stomach, intestine, and rectum. And then I just put these together to kind of help, again, to show these differences, right? So you have villi and crypts in the small intestine. You can see that here. You have these epithelial transitions happening at the beginning of the digestive tract in the esophagus, and then again at the end in the large intestine. And then in the stomach here, you can see the columnar epithelial cells. For lab exercise 7.3, this is a fetal pig dissection, and this is for in-lab students only, but you should still be able to, if you're an online student, be able to identify these structures and the figures that are to follow. You'll want to be able to identify the mouth, oral cavity, esophagus, stomach, small intestine, large intestine, liver, gallbladder, pancreas, rectum, and anus. So first here we've got, this is the mouth of a fetal pig, showing the hard palate, soft palate, the epiglottis. It's not very well shown in this image, but that is where the epiglottis and the glottis are. And this is the tongue. Next you have the esophagus here, larynx, trachea. And in the abdomen, you have the lungs here, which is just provided, they're just providing a frame of reference. What you want to label are the stomach, the pancreas. The liver is this large structure here. Gallbladder is going to be right underneath the liver. The duodenum is the start of the small intestine, which is this here. Duodenum, small intestine, and then large intestine. This is another view of the liver, showing the gallbladder behind it. There is a bile duct coming off of the gallbladder, and then again, small intestine, large intestine. Thank you guys for watching. Don't forget to do the reading. Check the description for more videos related to these topics, and leave your questions for me in the comments below.